Okay, so I know the the last video cut off pretty abruptly, so I just wanted to briefly kind of reconvene with this. Essentially, I showed two main take-home messages. Take-home message number one from all of this was that the gene expression levels, all right, and we can kind of see that here and here, gene expression levels of our iPS cells were similar to ES cells and no longer similar to fibroblasts, and same with protein expression. Right? And again, this was all derived by going back and forth between the results and the figure as I went along. Global gene expression also represented embryonic stem cells, and iPS cells were more similar than the iPS cells were to the original fibroblasts that they were derived from. And finally, that the exogenous, in other words, the retroviral extra copy of these four factors were turned off, but the genomic copies of these genes, so the endogenous ones, the ones that are part of the cell's own genome, were actually then turned on as part of the induced pluripotent stem cells. And that's going to be important so that they do not continually express the transgenes for continuous maintenance of pluripotent stem cells and self-renewal. This is the figure that I'm going to walk through as an example of what I expect you to do. So you guys eventually are going to explain figure 5 to each other as part of a discussion group, and you're going to create a video of yourself going through figure 5. In light of time, I kind of went through this figure a little bit faster, but there will be a demonstration of me going through this figure in the format that I'm looking for you as an example kind of in that discussion board. So look for that. In the meantime, again, I just want to keep kind of working our way through some of these figures and showing how I go back and forth between the results and the figures so that you can do that on your own. And so in light of time, I'll just keep moving pretty quickly though. Figure 3 here. Figure 3 is one of those that would be extremely difficult to understand if you just came at it blindly. Right? What do I have all these dots here for? I have a bunch of dots shown here, graphs, and even if I look at the figure legend, bisulfite genomic sequencing of the promoter regions, open and closed circles indicate unmethylated and methylated CpGs, luciferase assay driven by promoters. This would be pretty difficult to really understand, but remember, if I go back, I have an idea of what I'm looking for. I've already shown morphology. I've already seen the figures that demonstrate feeder dependence, surface markers, gene expression. Now I'm looking for the promoter activities. And I have this check mark here because this figure is what's going to do this. But looking for this, I want to see if the promoter activities are similar in my iPS cells to the embryonic stem cells and no longer similar to the fibroblasts. So if I briefly look up a little bit about methylation of CpGs, what I know is that methylation, all right, so methylation, if the circles are closed, is a gene that is off. So basically, if you think about this, this is developmental biology in a nutshell. But if the genes are wrapped around chromosomes, they are tightly packed away, and genes that are tightly packed away are not being expressed. All right, think about that old shirt in your closet that you never wear. It's tightly packed away in a box in the back of the closet. But the ones that you wear all the time that are in your regular rotation, they're going to be out in front and easily accessible. So unmethylated gene regions are ones that are very accessible, and methylated ones are unaccessible. So basically, if you see a lot of open circles, it means that these genes are on. If you see a lot of closed circles, it means the genes are off. And you can see that for the most part, the endogenous, this is the genomic, the cell's own copies of OCT34, REX1, and NANOG, they're pretty much off in the fibroblasts, but they are on in our induced pluripotent stem cells. So these genes have been turned on. This is the epigenetics. This is the promoters are on. And they can take the promoter region for these genes and put them just in front of a luciferase, which basically tells you if the gene is on. And again, you see that the genes are on, similar to our embryonic stem cell cell line, and they're not on in the original fibroblasts. So again, this is just saying that even the promoter is changing epigenetically in ways that make our induced pluripotent stem cells similar to embryonic stem cells and not the fibroblasts. All right, then figure 4. This is talking about high levels of telomerase activity. So if we're talking about telomerase activity, remember, telomeres are the ends of our chromosomes, and so those chromosomes, every time the cell replicates, gets chopped up a little bit and a little bit more. And so if a cell is immortalized like a stem cell would be, or unfortunately cancer cells, I've turned this on as well, then they have telomerase which extends those, right? So this is showing that there are telomerase activities in our induced pluripotent stem cells. All right, so our cell lines have telomerase activity, the entera embryonic stem cells have telomerase activity, the HDF do not. But if I'm really going to understand this, I need to understand this trap method. All right, so if this is where I would go to the methods and figure out what the heck they're doing here. All right, so in the methods, there's a section called detection of telomerase activity. Telomerase activity was detected with a trapeze telomerase detection kit, according to the manufacturer's instructions. All right, well, I need to look up that kit then. All right, so Chemicor actually is now a part of Millipore, but I can look up that kit, and in fact, I've done so. I'll show you in a second here. If we go to the web here, I can look up the trapeze telomerase kit. So this is the Millipore site. So on the Millipore site, I can get to the user guide. In that user guide, basically, if I go through it a little bit, has an introduction of what telomerases are and kind of a generality of the technique. Within 5 minutes, even if I had no clue what trapeze was to start with, just reading these 2 pages, I basically can get it down. And just another Google search starts to have a lot of images that really start to explain what's going on. So you can look up these techniques even if you start with no knowledge of them, and you can really learn what's going on. And by learning what's going on, you're going to be able to describe this even better. So let's take a look at this. This is just the information I got off the internet, again in that 5-minute search. Essentially, telomeres add these repeats. So these are the telomeres, and telomerase adds the repeats so that your telomeres grow the same extent they would normally shrink. And this is important if something has become a stem cell, because stem cells need to divide indefinitely. All the trapeze does is it creates this little template. This is basically like the end of a telomere on its own, and if telomerase is active, it will basically start adding these repeats to this little sequence that our kit has added. And by adding these repeats, this gets longer and longer and longer, based on these 6 nucleotides at a time. And so at the end of this, if telomerase is active, you're going to have extension of this by a lot of different random amounts. All right? That's what GGTTAG to the N is, meaning you can add as many as you want, or as many as the telomerase is able to in this time. And so then when you run this on the gel, these bands down here are the sequence just of this, where there was no extension, but we get larger and larger bands. Remember, small bands, small things go through the gel fastest, so it was loaded here, these went through the gel fastest. Large things go through the gel slower, and we can see that we have bands because there are different extents of the GGTTAG added. And so what that means is if you get these bands, kind of this ladder of bands, it means that telomerase was active, and if you don't, it wasn't. So if we look back here, we can see that in our human dermal fibroblasts, there is no telomerase activity. We don't see those bands. But in our entera cells, ES cells, and our induced pluripotent stem cells, we see those different bands. So telomerase has been turned on in these cells, even though that telomerase was off when those cells were fibroblasts, before we dedifferentiated them. Now the heat basically just kills the telomerase. So heat denatures proteins, so if you denature telomerase, you don't see those anymore. So it's just proving that this is indeed due to telomerase enzymatic activity that is inside the cell. And here's cell number, so basically you're looking at proliferation, and the cells are continuously growing and growing and growing, so they're dividing indefinitely. And so now we basically have shown, in addition to what we had before, we've shown telomerase activity and we've shown proliferation. So basically at this point we have shown that our induced pluripotent stem cells are looking like, acting like, behaving like, and expressing genes and proteins similar to embryonic stem cells. They are no longer fibroblasts, they're now stem cells. Now, what I'm going to have you do is I am going to have you discuss figure 5, and you're going to also talk about figure 7. Right? So if we look at figure 5 here, you guys are going to go through this, but I'll give you a quick hint. Remember, we've already shown these things. We need to expect these two. So what's left? Well, they need to show that their iPS cells differentiate. And if they're going to differentiate, they need to differentiate into endoderm, mesoderm, and ectoderm, because you have to differentiate into all three germ layers if you've made it a pluripotent stem cell. So you should expect to see things that are trying to prove that they can now differentiate. We've shown dedifferentiation. Now they need to show redifferentiation into multiple cell types. All right, so you're going to go through this. You're going to basically tell me what this means, and eventually you're going to walk the audience through this, and I'll give you examples with that in one of the later discussions for this one. And again, just remember, you can keep going back from the results to the figure. There's a lot of things that'll tell you what each of these things are. And then figure 7 you're going to go through as well. So that's what you two are, that's the two figures that you are all going to kind of go on. Hopefully you can figure that out by now. And so that's part of the last little bits of the activity worksheet that we've been going through. Then you should be able to submit that. And at that point you are ready for the discussion board. And then you will be ready to essentially walk your audience through figure 5 more directly, and then we have techniques left, and then you basically can finish your final analysis of the article for the end of the week. All right, good job everyone.